Imagine for just a second that you are tasked with building a video game. But not just any game. Your objective is to program a perfectly accurate living human cell.
Right, a total digital twin.
Exactly. Every single pixel, every floating molecule has to perfectly obey both the rigid laws of physics and the incredibly messy, chaotic rules of biology. Now, whether you study computational biology, work in medicine, or you just love the absolute bleeding edge of tech, this deep dive is going to fundamentally change how you visualize the inside of your own cells.
It really is a massive paradigm shift. I mean, for decades, the idea of a fully functional 4D digital twin of a cell just felt like, a pipe dream.
Yeah, totally impossible.
But we are finally looking at the realization of that concept. There's this fascinating paper published in July 2026 in the journal Cell by Arkveld and a huge team of researchers, and their mission was exactly what you just described. They managed to create a whole cell four-dimensional particle-based simulation using actual live cell microscopy data.
And just to set the stage for you listening, they didn't just pick a simple, easy-to-model bacterium. They used a human cancer cell line, specifically an oral squamous carcinoma, and they mapped its mitochondrial network.
Which is incredibly complex on its own.
Right. But before we get into the weeds of how they actually pulled this off, I want to back up a step. We've mapped the human genome. We have these gorgeous, high-resolution structural snapshots of individual proteins. So why has it been so incredibly hard to just put those pieces together and simulate a whole living cell in motion?
Well, it comes down to a classic observational catch-22. To build these whole cell models, or WCMs, scientists need incredibly precise structural data. Historically, to get that level of detail, you had to use techniques like cryo-electron tomography.
Which is where they essentially flash freeze the cell.
Yeah, right, exactly. You drop the temperature so fast that ice crystals don't even have time to form, which preserves the delicate structures. So you get a beautiful, incredibly detailed 3D snapshot. But there's an obvious catch. The cell is dead.
Right. Perfectly still.
It is completely static. You have the structure, but zero information on the dynamics, like how things actually move and interact in real time.
So if you want to see movement, you have to look at a living cell.
Yeah.
But I'm guessing that comes with its own set of problems.
The massive problems, yeah. If you wanted to see the living dynamics, the standard approach was confocal fluorescence microscopy. You tag the parts of the cell you want to see with fluorescent molecules, and then you just hit them with a laser to make them glow.
Okay, but if you're trying to capture a continuous 3D video of the entire cell over time, doesn't that mean you are constantly blasting it with highly concentrated laser light?
Yes, and that causes severe phototoxicity.
Right, phototoxicity. I always picture this like trying to film a bustling city highway at night, but your camera requires this massive blinding flash bulb. And eventually you just blind all the drivers, cause a massive pileup, and melt the asphalt.
That's a very accurate way to look at it. You just can't observe natural biological behavior if your observation tool is acting like the world's tiniest microwave and literally cooking the environment.
So what changed? How did they get around the microwave problem?
The breakthrough solution that actually provided the foundation for this digital twin is a technology called lattice light sheet microscopy, or LLSM.
Okay, LLSM. How does that work?
Instead of a blinding concentrated laser beam that penetrates the whole sample at once, LSM uses a razor-thin structured sheet of light. It gently sweeps this optical sheet through the cell, just layer by layer.
Oh, wow. So it's much faster.
Super fast. It captures 4D data, so X, Y, Z, and time, taking a full 3D frame of the entire cell every 11 seconds. And because the light is spread out into a thin sheet, the energy absorbed by any single part of the cell is minimal. It virtually eliminates the phototoxicity.
Giving us our first truly untainted look at live cellular dynamics at this scale. That's incredible. And the specific dynamic they chose to look at is the mitochondria. Now, if you're listening to this, you likely know the mitochondria as the powerhouse of the cell.
Right, the classic middle school biology definition.
Exactly. But we really have to forget the old textbook drawing of those static kidney bean-shaped batteries just floating around.
Yeah, that static image is incredibly misleading. In reality, mitochondria form these highly dynamic, sprawling, interconnected networks. They are constantly moving. They are involved in signaling, in managing the cell's calcium levels, and they dictate the life or death cycle of the cell.
And when these networks dysfunction, you see it implicated in severe diseases, right? Everything from cancer to diabetes to neurodegeneration.
Absolutely. And they're constantly remodeling themselves, which is the really tricky part to simulate. You have fusion, where multiple mitochondria physically merge together into a larger network.
So they merge to share resources.
Yes, to share resources, crank up ATP production, and even buffer against toxic spikes in calcium. And then, on the flip side, you have fission, where they pinch off and split apart.
Okay, so fusion is merging, fission is splitting. Why do they need to split?
Fission is crucial because it allows the cell to quarantine damaged mitochondrial DNA so it can be degraded. It also breaks the network down into smaller fragments that can be easily transported to different areas of the cell that might need an energy boost.
Okay, so let's use an image memory technique here for everyone listening, just to visualize this paper's actual data outputs. Imagine the cell's nucleus as this massive blue planet, and surrounding the whole thing is a red plasma membrane, like a force field.
I love that visualization.
And then the mitochondria are this sprawling, shifting green web of particles stretching between the blue planet and the red force field, constantly merging and splitting. So before we jump into how they turn that green web into computer code, let's just do a quick subtopic recap.
Great idea. So, bullet point 1. Whole cell models historically predict cell behavior, but lack dynamic live data because observation killed the cell.
Bullet point 2. LLSM, or lattice light sheet microscopy, solves this by providing 4D imaging with a razor-thin sheet of light so we don't fry the cell.
And bullet point 3, mitochondria are not static beings. They constantly remodel through fusion merging and fission splitting.
Which brings us to a question for you, the listener, to chew on. Think about a city highway system. If fusion is the highway merging to share traffic, what happens when there's an accident? That's right, fission acts as the detour to quarantine the problem. So what stands out to you about visualizing organelles as shifting highways rather than static objects?
It really changes your whole perspective on biology.
It really does. Okay, so we've got this beautiful 4D video of a biological highway system. But here's the fascinating problem. How do you actually translate that shifting green web into a computational physics engine? Like, how do you turn biology into math?
Well, to do it, they had to start by simplifying the system. You just can't model the whole complex machine at once. So they actually started by breaking the real cell.
Wait, they broke the cell on purpose.
They did. They treated the live cancer cells with a chemical called nucleus for 60 minutes.
But that seems entirely counterintuitive. If your ultimate goal is to study how a healthy living thing works, why would you build a model of a living system by poisoning it first?
It sounds backward, I know, but think about it like trying to reverse engineer the suspension system of a car you've never seen before. If the car is driving down the road with the engine revving, the driver steering, and the brakes engaging, there are just too many variables.
Oh, I see. You don't know if a sudden jolt was caused by a pothole or the driver slamming the brakes?
Exactly. So what do? You take the engine out entirely, you strip it down, put the car in neutral, and just push it down a hill. By watching how it rolls purely based on gravity, friction, and the passive physics of its suspension, you can build a baseline model.
That makes total sense. So in the cell, the nucleus drug is what takes the engine out.
Precisely. Mitochondria don't just swim on their own. They are actively pulled along microscopic protein tracks called microtubules. Nucleus chemically depolymerizes it. It dissolves those microtubule tracks.
It melts the roads.
Exactly. It completely strips away all the active motor-driven transport in the cell. So the mitochondria were left just passively floating, diffusing through the cytoplasm, purely based on thermal physics.
Okay, so we have our baseline. A cell with no active engines. How do we build a digital car to match it?
They used a highly advanced particle-based software called READY. First, they took those beautiful images from the microscope and ran them through a segmentation tool called mitograph.
Mitograph, got it.
Yeah, and this tool takes the blurry, glowing biological images and extracts a clean, mathematical 3D skeleton of the mitochondrial network.
And then the READY software populates that digital skeleton with particles. And we're talking extreme precision here. Each digital particle was coded to be exactly 0.15 micrometers in radius, which perfectly matches the biological width of real mitochondria.
Right down to the fraction of a micrometer.
But here's the part I really want to dig into. How do you program biological reactions like fusion and fission into a computer simulation? It's not like the computer knows what a mitochondrion is.
No, it only knows physics and geometry. In the software, these digital particles are connected to each other by what we call harmonic bonds.
So they're essentially digital springs holding the particles together in a chain.
Yes, exactly, digital springs. To simulate fission, the splitting of the mitochondria, the algorithm runs a stochastic process.
Meaning random.
Random, but governed by strict probabilities. The researchers experimentally measured how often these splits happen in real life. Then they programmed the digital springs to have a very specific probability of suddenly breaking. In this case, the rate was exactly 0.00044 times per second.
Wow, that is specific. 0.000044 per second. And what about fusion? How do two separate digital chains know when to merge?
Fusion is programmed as a distance-dependent topological reaction. The software tracks the position of every single particle using something called overdamped Langevin dynamics.
Okay, hold on, stop right there. Overdamped Langevin dynamics. We definitely need to translate that for the audience.
Fair enough. Imagine you are blindfolded and trying to walk across a crowded, muddy mosh pit at a concert.
Sounds terrifying, but okay.
Right? You aren't moving smoothly. You are constantly bumping into things, experiencing friction, and getting randomly shoved around by the crowd. That's exactly what particles experience in the thick, viscous cytoplasm of a cell. Overdamped Langevin dynamics is just the math that calculates exactly how things randomly wander around in that kind of thick, fluid environment.
Got it. So the computer is calculating this muddy mosh pit math for every single particle simultaneously.
Right. And if two separate mitochondrial particles randomly bump into each other in that mosh pit and their centers fall within a specific reaction radius, the computer instantly forms a new harmonic bond, a new spring between them. They calibrated this fusion rate to 0.1 per second.
And they did all this to make sure that the digital network maintained the exact same average fragment length as the real nocodazole-treated cell they were watching under the microscope.
Exactly. And this is where we see if the baseline actually works. They measured the mean squared displacement, or MSD.
Which is basically just a fancy mathematical way of measuring how far a specific particle manages to wander away from its starting point over a set amount of time.
Right. And when they compared the MSD of the digital twin to the MSD of the live biological data, it was identical.
That's incredible. Like statistically identical.
It was a perfect match. The temperatures were locked at 37 degrees Celsius, the fluid viscosities matched, the random particle collisions matched. By successfully matching the mean squared displacement, they proved beyond a shadow of a doubt that their underlying digital physics engine was completely sound.
Okay, so let's do our next 5-minute recap of where we are. Bullet point 1. Nocodazole was used to dissolve the microtubule highways, creating a baseline passive model with no active engines.
Bullet point 2. The digital twin was built in the READY software using 0.15 micrometer particles connected by digital springs.
And bullet point 3. Reactions like fusion and fission were coded purely as the breaking and forming of those harmonic bonds, matching the real-world mean square displacement.
Which brings us to my question for you listening. By successfully matching the MSD, we proved our digital physics engine works. But life isn't passive. How do you think the system changes when we turn the engine back on?
I love this part. We're upgrading to the active twin. We're looking at healthy, untreated control cells now. And this introduces a whole new layer of complexity. We have to add those microtubule tracks back into the simulation.
To do this, the researchers developed a custom software tool they called ready-made models, or RMM. RMM read the 3D imaging data of the healthy cells and procedurally generated the full microtubule network right inside the simulation. Let's add them as magenta train tracks to our visual memory map.
Blue planet, red force field, green web, and now magenta train tracks.
Perfect. But it's important to understand that microtubules aren't just dumb, empty pipes. They are highly organized, polarized structures.
Meaning they have a distinct direction, like a one-way street.
Exactly. They generally radiate outward from the center of the cell, near the nucleus, pushing out toward the outer edges of the plasma membrane. And to navigate these directional streets, the cell uses biological motor proteins that latch onto the mitochondria and literally drag them along the tracks.
I actually have a mnemonic for you listening to help remember how these motor proteins work. It's KADR. Kinesin and dynein are the two main motors. Kinesin is anterograde, meaning it steps outward toward the cell edge. Dynein is retrograde, meaning it steps backward, inward toward the nucleus. So KADR, kinesin anterograde, dynein retrograde.
That is a brilliant way to remember it. So the researchers had to introduce these motors, the kinesin and dynein, into the simulation. They coded them as two-state Markov models.
Let's demystify that term, too. A Markov model is just a mathematical way of dealing with probabilities over time, right?
Exactly. Think of it like a flickering light bulb that randomly switches between being on and off. At any given millisecond in the computer simulation, a digital motor has a statistical chance to switch from an inactive state to an active state. When it becomes active, it physically latches onto the microtubule track and starts pulling the mitochondrial particles.
And it isn't an unbreakable grip either. The simulation includes force-dependent detachment. If a motor is pulling a piece of mitochondria, but it gets snagged or the tension gets too high, the motor will literally snap off the track so the whole system doesn't break.
It's incredibly detailed physics. And because the mitochondria are now being actively yanked around the cell by these motors at high speeds, rather than just drifting lazily, they are crashing into each other way more often.
Right. If cars are driving 60 miles an hour instead of just rolling down a hill, you're obviously going to have more collisions.
Precisely. So to keep the digital network from just fusing into one giant unstable blob due to all these new high-speed collisions, they had to tune down that intrinsic fusion parameter we talked about earlier. They dropped it from 0.1 to 0.047 per second.
Okay, so we've got the magenta tracks, we've got the KADR motors switching on and off, we've got the adjusted fusion rates. This is where the model really has to prove its worth, because honestly, anyone can build a model that mimics past data by just tweaking the dials until it matches. The true test of a mathematical model is prediction.
And this is where the paper transitions from being a cool descriptive tool to an absolute predictive powerhouse. The researchers set up a brilliant validation test. They took a completely new cancer cell, but this time they didn't treat it with nocodazole for 60 minutes to destroy all the tracks, and they didn't leave it completely healthy. They treated it with nocodazole for exactly 30 minutes.
So it's caught in this messy intermediate state. The microtubule tracks are only half destroyed. It's a biological gray area.
Exactly. They imaged this half-destroyed network under the microscope, extracted its specific broken geometry, and loaded those specific tracks into the simulation. And then, and this is the really crucial part, they ran the active twin simulation without changing a single kinetic parameter from the healthy control cell.
Wait, really? They didn't adjust the motor speeds or the fusion rates to account for the drug at all?
Not at all. They kept the exact same mathematical rules, same motor probabilities, same fusion rates, same fission rates. They just placed those rules into the new half-broken environment.
And what happened? Did it break?
The digital twin perfectly predicted the behavior of the real cell. It accurately forecasted the new intermediate motility of the mitochondria. It perfectly predicted the shifts in the fusion and fission dynamics.
That is genuinely astonishing. The computer didn't just copy past data. Based purely on the laws of physics, the geometry of the tracks, and the biological rules programmed into it, it accurately predicted how a living cell would react to a half dose of a drug.
That is the absolute gold standard of a mechanistic model in systems biology. When your model can predict the outcome of a novel perturbation without you having to retune the underlying math, it proves that the rules of your simulation are fundamentally aligned with the reality of the biological system.
Okay, let's pause and appreciate what we just covered with another quick recap. Bullet point 1. Microtubules or magenta tracks and molecular motors were added for active transport.
Bullet point 2. Remember, KADR Kinson moves outward, Dynein moves inward.
And bullet point 3. The model successfully predicted the effects of a 30-minute partial drug perturbation without any retuning of the parameters.
So here's a question for the audience. If a computer model can predict how a cell reacts to half a dose of a drug just based on physics and geometry, what could this mean for the future of personalized medicine and drug testing?
It's wild to think about, which perfectly sets the stage for the final and arguably most important part of their research. Now that they knew they had this predictive powerhouse, they aimed it at a major biological mystery observed in clinical pathology, a phenomenon known as paranuclear clustering.
This is where things get really fascinating.
Let's break down what that means for a patient. Why do we care about paranuclear clustering?
Because it is a severe stress response. When a cell experiences extreme, life-threatening stress, for example, severe hypoxia where it's starving for oxygen, or during a viral infection like hepatitis B, or incredibly, when ovarian cancer cells develop resistance to chemotherapy drugs, the cell hits a panic button.
And what happens to the mitochondria?
They completely abandon the outer edges of the cell. They rush inward and cluster tightly together in a dense ring right around the nucleus.
A total retreat to the center. I assume this is because the nucleus is where the DNA is, and during high stress, the cell needs massive amounts of APP energy right there to transcribe emergency survival genes.
That is the prevailing thought, yes. The mitochondria relocate to where the energy is most desperately needed. But the scientific mystery has always been how the cell achieves this sudden, massive reorganization. The existing canonical literature theorized that it was purely a chemical signal. The theory was that the cell chemically alters the motor proteins. It dials up dynein, the inward pulling motor, and simultaneously suppresses kinesin, the outward pulling motor.
Basically putting all the engines in reverse.
Exactly. So to test this canonical theory, the researchers plugged these stress parameters into the digital twin. They altered the binding ratio of dynein to kinesin to 10-to-1. They increased the probability of dynein activation tenfold and dropped kinesin tenfold.
And let me guess, the simulation did exactly what the textbook said it would.
It did. In the simulation, the mitochondria rapidly aggregated around the nucleus. The average distance from the nuclear boundary shrank dramatically, from 6.6 micrometers down to 4.19 micrometers. The canonical chemical theory worked perfectly in the model.
But if I know computational biologists, they rarely stop at just confirming the textbook.
Yeah.
This is where you can do things in a computer that you could never, ever do in a wet lab.
Precisely. In a real living cell, you can't manipulate the physical tracks without also screwing up the chemical signals or just entirely destroying the integrity of the cell. But in a digital twin, you are God. So they ran a virtual experiment. They took that exact same high-stress environment, the exact same aggressive dynein motors desperately trying to pull the mitochondria inward, but they virtually snipped every single microtubule track perfectly in half.
Wait, explain the mechanics of that.
Yep.
If they snip them in half, do they remove half the material from the simulation?
No, and that's the genius of it. The density of the tracks remained identical. The total mass of the microtubule network in the cell was exactly the same. The chemical kinetic signals driving the motors were identical. The only thing that changed was the topology.
It's like taking a long, continuous highway that leads straight to the center of the city and chopping it up into a dozen disconnected, dead-end streets. There is still the exact same amount of pavement, but you just can't get to the destination.
Exactly. And the result of this virtual snipping was profound. The clustering completely stopped. The mitochondria were left stranded out in the periphery of the cell. Despite the fact that the chemical signals were screaming at the motors to pull inward, they just couldn't do it.
Because the bridge was out. It doesn't matter if you have a Ferrari engine under the hood and your foot is stomping the gas pedal. If the highway doesn't physically connect to the destination, you aren't getting there.
Which reveals the core insight of this entire paper. The physical topology of the microtubule network acts as a structural gatekeeper. It is completely capable of overriding the cell's chemical kinetic signals. The global physical architecture is a hidden control variable.
That is a massive revelation. It means that the physical skeleton of the cell dictates what the chemical signals are actually allowed to achieve.
It explains so many inconsistencies in clinical lab results. Sometimes oncologists or researchers will measure the expression of motor proteins in a biopsy and assume they know how the organelles will behave. But this proves that measuring the chemical signals isn't enough. If you don't know the physical topology of the cell's highway system, you don't actually know what the cell is capable of doing.
Okay, let's tie this all together with our final recap. Bullet point 1. Stress causes mitochondria to cluster near the nucleus via dynein upregulation.
Bullet point 2, virtual halving the microtubule lengths completely aborted this clustering, even with the stress signals active.
And bullet point 3, microtubule topology is a hidden control variable for organelle distribution.
So my final question to you listening, if you were designing a cancer drug to stop this clustering, would you just target the motor proteins, or would you have to look at the structural integrity of the cell's highways as well?
Wow. We have covered a massive amount of ground today. We went from shining a gentle sheet of light on a living cancer cell to building a 4D digital twin that literally proved that the physical layout of a cell skeleton can override its chemical signals.
And we really must emphasize why the specific mesoscale approach is such a leap forward for computational biology. Whole cell models that try to track the quantum mechanics of every single individual atom are just computationally paralyzing. They take supercomputers months to simulate milliseconds of time.
Which is useless for rapid drug testing.
Completely useless. But this mesoscale model tracking physical shapes, network topologies, and basic reaction probabilities is incredibly lightweight and efficient. Simulating 2 full minutes of these complex biological events only took about 14 minutes of processing time on a standard desktop workstation. It is efficient, it is interpretable, and it is highly predictive.
We started today by imagining a video game of a living cell. Arkfeld and his team haven't just imagined it. They've effectively built the physics engine for that game. So I want to leave you listening with this final thought. We've just seen how a computer can use the physical architecture of a single cell to accurately predict how its mitochondria will react to a drug. But what happens tomorrow? What happens when researchers add the endoplasmic reticulum, the Golgi apparatus, and full genetic transcription circuits into the software? If we can eventually simulate how your specific unique cells with your specific physical architecture will respond to a pharmaceutical intervention in a computer before you ever take a single pill, how close are we to a world where trial and error medicine is completely obsolete? Ponder that. Thank you for taking this deep dive with us, and as always, keep exploring the incredible machinery of life.